I share my screen or?
Yes, you can. So, my dear student, colleagues, and all the viewers who are watching this program live from Facebook page, Physics Adda and School of Astrophysics and Astronomy, and YouTube channel Pretam Kumar Das, I'd like to welcome you all to our international physics webinar. So, good evening to all here in Bangladesh, and a very good morning to all those who are watching this program live from USA. So, hope you are well and safe from Corona pandemic. So, today it's our 156th International Physics Webinar. So as we know that it's very difficult for us to continue our normal academic program inside the campus due to the Corona pandemic. So we have to start our online program. I think you have already come to know that the Department of Physics, Pabna University of Science and Technology has started its online program including International Physics Webinar, and we have successfully completed our 155th International Physics Webinar, including two Nobel laureate speakers. And today is a very special day for our department. Today I'd like to welcome you all to a joint session between the Department of Physics, Pabna University of Science and Technology, and the Department of Computational and System Biology, School of Medicine, and the Department of Physics and Astronomy, University of Pittsburgh, USA. And we have with us here today Dr. Jianhua Xing, Associate Professor, Department of Computational and System Biology, School of Medicine, and Department of Physics and Astronomy, University of Pittsburgh, USA. And he has already connected with us, so I'd like to welcome our speaker, sir. Good morning and good evening to here. Thanks for accepting our invitation, sir. And we
Thank
would
you.
like to say thanks on the behalf of the Department of Physics, Pabna University of Science and Technology, for accepting our invitation. So I think you have already come to know the title of this today's International Physics webinar, and the title is Studying Cell Phenotypic Transition Dynamics from a Chemical Physics Perspective. And our speaker is Dr. Jianhua Jing, Associate Professor, Department of Computational and System Biology, School of Medicine, and Department of Physics and Astronomy, University of Pittsburgh. So before going to our speaker, I'd like to introduce our speaker with all of you, and we have divided uh, our webinar into three parts. First of all, we'll introduce our speaker with all of you, and then our speaker will deliver his speech, and at the end we have a discussion time. In that time, anybody can join with us, and you can ask question by commenting also. So we can see Dr. Jianhua Jing completed his middle school in Beijing and high school also in Beijing, and bachelor in chemistry in Peking University and masters in chemical physics, University of Minnesota, and the PhD from the University of California, Berkeley. And we can see his postdoc. He worked as a postdoctoral fellow researcher at the University of California and PBI fellow at the Lawrence Livermore National Laboratory. And he started his as an assistant professor in Virginia Tech in 2007, and he stayed there up to 2014. And then he joined at the University of Pittsburgh as a assistant professor in 2015, and now he is working as an assistant professor at the same university, School of Medicine. And we can see his field of research. The Jing lab is interested in the following fundamental questions: how do thousands of molecule species orchestrate temporally and spatially to determine a cell phenotype? How can one regulate the direct cell phenotype? Specifically, the lab currently focuses on epithelial to mesenchymal transition (EMT), characterized by loss of cell-to-cell adhesion and increased cell motility. EMT plays important roles in embryonic development, tissue regeneration, wound healing, and pathological processes such as fibrosis in lung, liver, and kidney, and cancer metastasis. So lab studies the coupled gene expression and epigenetic dynamic of EMT. We can see his Google Scholar ID. His citation is more than 2700, and his research ID is his research point is 36.35. So this is a recent publication. So thanks for all of your patience. Now it's time to go to our speaker. Sir, I'd like to welcome you again. Sir, it's your time, sir. You can start your session, sir.
Thank you, Pretam, for this introduction.
It's my
Um,
pleasure, sir.
yeah, thank you. And I'm really happy to be here, and especially at a difficult time. First of all, I want to want to say thanks to Pat, and also I hope everyone's in the safe situation now. I think that now it's definitely it's a difficult situation, but things will definitely about change. Let's just hope for the best. So with that, let's see. Today I want to share with you guys some of our research on cell phenotypic transition dynamics. So what I do is I'll first give some background, then I'll talk about our work on lifestyle imaging. Then there's another work we try to reconstruct vector field from single cell genomic data. Suddenly I'll give a summary and acknowledgement. So let's go back to background. I know the students from physics department. So, but the basic biology like this, we know in our body there are many different cell types, as shown here. They're very different, and they clearly have different function, different forms. But we know that they all come from the same set of genome, right? They have the same set of genome. They come from one fertilized egg, right? So this is a fascinating question: how one fertilized egg can develop into different cell types. So as shown here is embryo, right? Then in this process, specifically, Pretam just mentioned this EMT, and its reverse MET play a very important role during development. That's how cells can migrate to different places, then develop into different tissue and organs. And also to be hijacked by pathological process like cancer metastasis. So show is a solid tumor, and their cells can kind of detach from primary tumor, go to the circulatory system, then eventually leave this circulatory system, undergo a reverse MET, become develop into secondary tumor. So, and you might have heard this thing, several years ago. There's Nobel Prize to give to Kia Yamanaka. For his work, that we have the terminal differential cells. Usually we say the developmental processes from a fertilized egg, you develop into different cell types. It's kind of one directional. But actually, he showed you can really reprogram cells, terminal differential cells, into stem-like cells, then induced to other cell types like cardiomyocytes, heart muscle cells, or you can even directly convert from one cell type to another cell type. So you see, this pathological process, this cell phenotypic transition is ubiquitous in biology, and it's very important for a developmental process, and also many pathological processes involve cell phenotypic dysregulation. Therefore, you see, there's a strong need that we want to control the cell phenotype. We want to know how the cell control their phenotype, and also how we can control them. So as Pretam just mentioned, my background in chemical physics. So my PhD studies chemical reactions. So there, I show you is a kind of prototype in the field where time goes the hydrogen molecule plus hydrogen atom go to hydrogen to hydrogen. So hydrogen exchange, right? So this seems very boring, but it's the basic for chemical reaction, just like a hydrogen atom for atomic physics, right? So this is show that we have the potential diagram, and you can study it. This is later now. So we're talking about the proteins, but all this we're talking about is a kind of femto to millisecond scale, angstrom to nanometers. This is the typical time scale and size we'll talk about. Here, talk about the cell biology. This is what I'm currently doing. So I'm talking about the cell. It's kind of for talk about mammalian cells, many microns. And then there are many organelles. Then, the cellular process we're talking about is a millisecond to weeks, even longer, right? And the size, I said, is kind of micron or even millimeter or even larger for tissues. Clearly, you see there seems quite different field. So one is what I got trained, and the other one is what I'm doing right now. However, if you think about it as a physicist, right? You think, see, actually there are some similarities between the two, this macromolecular dynamics and cell dynamics. But at the end, they are dynamical systems. So as a physicist, you can write down a mathematical equation to describe this dynamics. Then you see there's common language. So actually, I many years ago I have done some work, theoretical work, to show even for a system governed by this Langevin equation. I don't know if you have a touch on this, but don't worry about this. Now it's just mainly a system, undergo some dynamics, then there's some noise. So it's a very generic form to describe the dynamics, thrown by some bus, right? You can think about that. Then I can show it's really can map to a Hamiltonian system. There, I can, one can utilize some of the ideas we are familiar with in equilibrium system, closed equilibrium system, to study this non-equilibrium process. So I won't go that in detail, but just want to mention this. There's a theoretical basis for this, right? Okay, so now they come back to chemical reactions. If you have ever taken chemistry, introductory chemistry, chemical dynamics, you may have heard this. For chemical reaction, you may think about, it's like energy profile. That you have reactant, you have product, you have energy barrier. So then it's along one-dimensional reaction coordinate. So kind of way we describe the dynamics. Or you can go to a little higher dimension. So, the system governed by some potential energy surface. So you can, classical system, which is a Newton's equation. Then if you may have some quantum effect, but at the end is the idea is you have a kind of a potential energy surface that this govern, the system move along this surface. You may have some attractors corresponding to stable conformation. Then there's a transition between them, right? So you have a lot of language. We talk about the reaction coordinates I just mentioned. Then there's other like reactive fluxes, transition path sampling. So this is very it's a central topic in chemical physics, starting in 19th century. Okay, so this is this is the chemical physics. Then let's talk about the cell biology. As I mentioned, seems there's kind of a similarity between the cell system and the molecular system. And here, there's some unique thing is nowadays. So I don't know if you know molecular physics. There's important technique is we can observe single molecule, then see how it moves, right? That single molecule technology gives a lot of information. Similarly, in cellular system, we can study single cells. So I show you some papers. So you don't need to worry about detail. The main thing is people here, the cell definitely not the atom, this is a coordinate, but rather the gene expression level. So people can really monitor, really study, right, to measure each individual species, molecular species as level. So this gives us kind of very important information. Just like in molecular system, we have X-ray structure, we have cryo-EM, we give the atomic, the structure at atomic resolution. So here give us the expression levels, but definitely also like the cryo-EM thing is a kind of a snapshot. So therefore, the limitation is we have all those existing high throughput masses, then give us the snapshot system information of the system, but we do not have the temporal information. When we study a system, we want to study the dynamics, right? So in order to do that, we have to do live cell imaging, really trace individual cells over time, just like we introduce individual molecules over time, right? So, but there are some difficulties. So this is a kind of a challenge my lab trying to deal with and discuss today. So here, I want to point out one thing. I know you guys are physicists, so nowadays cell biology or biology in general get into a very exciting field because all the techniques generate a lot of data. So I think this is kind of a parallel to what happens in physics several centuries ago. If you, as I show you, this is the movie, right? You see, this is the solar system. All those planet moves, right? So what happens in physics, astronomy is, you may remember this Tycho Brahe. He spent all his life just observe all those planets, records their position, right? Then his student Kepler, analyzing the data, he found some important rules, right, of the planet motion, and those eventually lead to Newton. Newton generates universal gravitational theorem, so form this framework of Newton's dynamics. So this becomes really a very great achievement in physics, right? So that's really makes physics beyond just data science. Now it's not just analysis, but now we have the theory. Then we coherent the theory, we can also predict things. For example, I show you, this is you see, I talk about the planet. Here is a protein, that SARS-CoV-2 virus protein. And there you see this is the result from molecular dynamic simulation. So each residue, we assume they follow the Newton's dynamics. Then you run simulations, you see this. So you see, although the theory can describe both the planet motion and this molecular level thing. Actually, I want to mention a few years ago, 3 researchers received a Nobel Prize in chemistry because they can further show you can utilize those kind of modeling methods, study a chemical system like this. Okay, so with this, I ask question. Yeah, now we have lots of data for cell. Can we do something like that? Can we find out the governing equations of cells? So this has been inspired me for a long time to think about it. Let's look at this very prototype. Let's EMT. Okay. So what I'm showing you here is the cell. So you see, this is cell. The human A549 cells, the lung cancer cell. So this is the original, right? Then we treated the TGF beta for 2 days. TGF beta is you can think about it as a molecule, right? Can cells take that molecule? It's got stimulated. You see, can induce to this kind of transition. You see, distinct different. And also during process, its molecular character also very different. So the gene expression. So what I show here is not A549, but another cell, MCF10A cell line, similarly undergo EMT. What I show here is you have you can see is this. So each row is a gene, and this is the time, right? You can see there's a dramatic change of the gene expression. So this is what physicists, chemists, biologists, they look at this. For physicists, as I mentioned, we are interested in the dynamics. We want to see some dynamic equations. How can we do that? So here's I'm thinking about, the first step is we should do what Brahe does, right? Get the kinematics. That is, can we simply use mathematics to describe its change? So how we do this becomes our the first topic I talk about is lifestyle imaging. The idea is we know for physical system, particle. So what we do is we have a coordinate, like Cartesian coordinates or action angle, whatever, and then we can use the coordinate to represent the state of the particle at a given time. Then you can work out the trajectory, right? So there this is kinematic, so we can further study what's the mechanism that become the mechanics, right? So we want to do the similar thing for a cell. So we want to know what X is, right? So first we want to know, also we want to experimental study. We want to monitor the cells over time. So there are a lot of constraints because cell, we know, is very delicate. You need to make the cell healthy, right? To stay healthy for a long time, several days, let's say. So therefore we have the constraint. We want to have a lifestyle imaging. We want to choose a coordinate that can really faithfully reflect the course of the transformation, right? And we want to, some quantity that are easily measurable, want to multiplex. So the more information get from system, the better understanding of the system. Then we need one minimal intrusive, minimal damage to cells. When we environment always perturb the cell, so we does not want to kill the cells. So first natural choice, okay, you have this molecule, molecular species come out the expression level, just like the when we do the gene expression. So there, people have done it. You can fuse a fluorescence protein to a molecular species that you can monitor. That's we know can result in had led to several Nobel Prize, if you know, for the fluorescence protein, and also to generally use the CRISPR technique. The things. So my lab has tried. It's not easy. It very time consuming. So we have a lot of limitation. Then we realize, okay, we want to have this dynamic equation. Okay. So but what is X? We have no constraint, right? So as long as there's something that can reflect the system dynamics, we realize one important thing. So one cell undergoes a morphological phenotype change, usually accompanies very large morphological change. If you remember my first slide, I show you different cells. You can clearly tell them, the difference, their shape, everything different. Therefore, we see, okay, we can measure those kind of features, which are easily to measure, right? For just transmission light, I think. Then we can use that. So this has been something have been recognized by many researchers, utilized to characterize those cell types. And especially, I want to mention, a colleague in India, because I noticed he has some, a nice work along this line. So important thing here, I want to point out, different from many other researchers. Instead of using this is statistical property, we use it's a dynamic variable because we want to study a dynamic equation. We treat a dynamic variable. So what we do is, like this human A549 cell line, I mentioned, there's a significant, noticeable morphological change, and also for this cell line we did use CRISPR to fuse a fluorescent protein to one of the important protein called vimentin. So there's a red fluorescent protein. Therefore, we can also monitor this protein. It's both expression and also it's a distribution, so-called texture features. Therefore, the key thing is you want to have mathematical representation is you have all the information. You need to quantify, digitize them into some quantity, right? So there you got to end up a vector. That's a vector represent the cell state. So this is what we did. Specifically, what we do is first step. Definitely, we need to quantify this the morphological features. We need a segment, right? We need because we have a lot of images, and we cannot just do by hand. We need a computer to do it automatically. Okay, here this is one cell, this is another cell. What the boundary look like? So we use deep learning. We developed a convolutional neural network algorithm help us to train the computer. So then the computer learn, then will do it for us. So this is published in this paper. If you're interested, you can look at it. Then that come to the pipeline, the framework we do is. That is, first we image the cells, then we segment, use that machine learning technique. Then we want to quantify, digitize. How we do that? For the cell shape, you see, I just we use a representative called the active shape model used in computer vision. So what to do is, for the shape, we place some dots evenly along the contour. Then, so each, say, we're talking about the two-dimensional here. I talk about a two-dimensional image. So each dot have this XY coordinate. Then that we already, by the way, I forgot to mention, first we have to align the cells. Then we can do this, okay? So each cell, we have this, each dot x, y coordinate. So each cell have this have n points. Then we have a 2n dimensional vector. So that represents the shape, approximate shape. So we can even do further some principal mode analysis. Essentially, what we do is we form a complete orthonormal basis set. So then any shape can represent the linear combination of this basis mode, basis vector. You can see that. So then you see this. If this you may recognize, oh, this is something we routinely do in physics, right? So now you see we have a representation. So each cell trajectory, each cell cell movement, that's a change will represent a trajectory in this shape space. So yeah, this is another shows how the overall procedure we do is we do time lapse, we do segmentation, we mathematically represent the cell state, and so extract some feature. I just mentioned the morphological feature. Similar for the Wilmington, we also have its texture. We have similar thing. We do certain quantification, get a vector. Okay, so we have a composite space, both for the morphological feature and the Wilmington texture feature. Then the cell represents this high dimensional space. So then for each individual cell, if we trace, we got the trajectory. You see, this is to become a trajectory. Then we can do analysis, just we do to analyzing a molecular, a molecule, the trajectory of molecule, right? So this, so there we have all the theories in chemical physics. Like here, I think about the system. You have we will have attractors corresponding to reactant product. So you're starting from reactant region. Then the cell can start to move. Sometimes it's leave this region, that's reactant region, then come back. So then, some attempt to make a transition, but failed, right? But then there are also possibility they can just end up into another attractor. So there's a successful transition. So they form different ensembles of trajectories. Then there are a lot of studies how we can analyze those trajectories. So this is an important problem in chemical physics and mathematical physics. So very interesting because I'm a chemist by training, and we really want to monitor how cell change, how molecule undergo a transition, which is technically very challenging. But now, because the cell is much bigger and the dynamics much slower, even me in my lab, I was trained as a theoretician, but now I have my wet lab, so we can monitor those transitions in high dimension, record those trajectories. Then we can do all those analysis. So I show you example. So this is one example cell. You see, the yellow is the Wilmington, and you see the cell. It's a 2-day long trajectory, and the cell undergoes shape. And also to see the Wilmington change a lot. So we have that data here. The right one, first one is the shell shape we tracked, and also the bottom one is the single cell trajectory in this morphological feature and Wilmington feature space, because of very high dimensions. So here only show you the leading to principal mode, but you can see the cell starting from one region, then move around, then become change to another phenotype. Yeah, we record many trajectories, and here show you two typical ones, but you can see they're very different, right? So all started from here, this region. So this one first undergo this see the Wilmington change first, then the morphology change. But this one, you see the Wilmington morphology, they change together. Indeed, we have many trajectories, so we can analyze. We do cluster based on how similar they are. then we can really class them into two groups, just similar to what I showed, the two typical ones. So yeah, with the trajectory, then I just mentioned we can do all this analysis. So this one is what do we do? It's in chemical physics. We know in this high dimensional space, a transit happens quite often. It's not through, from here to here, right? You can have through many, all possible paths, but different paths have different weight. So this is called path integral, right? And then most paths, most transit paths really concentrate on a small region. So we can, characterize those regions and see what's the dominant path. That's where to go, right? If you go to a mountain, you don't go to all the region. There are some saddle regions. It's easier for you to go than you go through that, right? Mostly go through it. Similar here, right? So you can characterize how it moves. You can define its reaction coordinate. So that's we use a technique called finite temperature string method, mainly trying to find the most likely path the system goes through. Then as show you see here example, we got that trajectory, the path, and all the trajectories essentially just fluctuate around, just like you would expect from this path integral picture, right? But interesting thing is for this system I just mentioned, right? This is two paths, so we can identify the two different paths. So this is very new for this system. Before people use the snapshot data, this genomic data, single cell genomic data, and they since you lose the temperature, the information about the dynamics, how individual cell move from one region to another, because you can only get snapshot. There, the data cannot really show this two path information, while the lifestyle imaging can do it, and you can even do further analysis. Let's see. Okay. This is high dimensional dynamics, but since it's most concentrate along a tube, so we may just write down effective one-dimensional dynamic equation I show here along this reaction coordinate. We take ansatz like this. You have move along one dimension. Since it's first of all, I want to mention this is a non-equilibrium system. So we know it's not a gradient system, right? It's not the dynamics governed by a gradient of potential minus gradient of potential, right? But it's a one dimension. You can since now we talk about this one-dimensional projection, we can define effective potential. So there we see you can even from the data, we can, because we record all the data, we have this dsdt. Then you do this average over all the trajectories. You will end up, got this dphi ds, because we assume the noise is have a mean, zero mean, uncorrelated, right? Then you can reconstruct this pseudo potential. It's not the real one. So this will show you the result along path. So you see, essentially tell you, if you from physics perspective to understand how this EMT, it will like this. You have this potential. The cell have initially trapped in this attractor. Then, but TJ beta, you clearly destabilize this attractor. So then the system relax to a new attractor. So this is what you may think about. You may remember if you have the atomic physics, right? The ground state, excited state, then relax. So here you see some similar language. Okay, yeah. For the biologist, they also want to use a kind of metaphor called avoiding landscape. They view developmental process like a potential, a ball sliding along the potential. Then at a certain point, you go here or go there. You go to different cell type. But here we really from the data we talk about how we can get this pseudo potential. Okay. So I think the question will be asked afterwards, right? So I'll just continue. So I talked about this lifetime imaging. Then we also have efforts on how can we understand the dynamics from this snapshot data. The reason is this, at the end, the biologists, they are interested in those genes, right? How the genes interact with each other. So so roughly speaking, if we want to understand dynamic system, we want to know, okay, a cell, where, what its current state is, and we want to predict where they go next, right? This is how we understand. So people have done a lot of efforts to use mathematical modeling to help understand, because the genes interact with each other. So mathematics can help us to understand. Let's say what I show here. I have two genes, X1, X2. They inhibit each other, right? They are not friends. They inhibit each other. And but they also have kind of self-activate each other, right? The more it has, and the more it try to generate itself. So this is this is based on biology. Then we can write down some mathematical equation to reflect this biology. So this is how we do it, right? You write down the mathematical equation, then the mathematical equation will describe how the system evolve over time. And the X1, X2, mainly just the gene expression, then we can measure experimentally. And dx dt actually form a vector field, right? Because I have two components here, it's become a field. Means along this X1, X2, any point, I have a vector. That's dx1 dt, dx2 dt. It's a vector, I show the arrow. It tells me where and how fast my system will go next. So, we know for this system, for Newton's system, we need to know both the coordinate and the momentum, right? But for this one, it's the first order, so we only need to know the derivative of the x. Then its dynamics supposed to be determined if we know the vector field, right? Well, here, if I have two genes, I done a lot of experiments, how me determine all those function forms, all the parameters, that's fine. But the cell have many genes interact with each other. Can we reconstruct a genome-wide vector field? And this seems impossible, right? Because there's so many genes interact with each other. How can you get all the information? And interestingly, then comes this work. This is several years ago. This researcher, they show, okay, when you measure this, what you do is destroy the cell. You have to kill the cell, then do the measurement. But this guy said, oh, actually, you got some information on the dynamics. You got some instant velocity. What that is, if you have any, know something about the biology, gene expression, that's like this. I can explain easily. So you have a gene. What the gene does is a DNA. DNA first, there's a transcription. So transcription become into mRNA. So mRNA, you see this called unsplice. You see there's some extra components. They undergo a process called splicing. Remove those extra components. Then this spliced mRNA becomes mature, then can be translated, become to protein. But eventually, those protein spliced mRNA will be degraded. So from a dynamic perspective, okay, for each process, you can assume there's a rate process, and you can write down some dynamic equation, some rate constant. Then those people say, oh, I can do the measurements. I can measure and supply the supplies. This can read from my data. And then, but there's a rate constant, which you can find some way to estimate. Then, if you can do that, then you have U, you have S, you have gamma. Then I can, estimate the dSdt. So this instant velocity, right? You can score that. So that's very inspiring work. So I thought, okay, now I have my dxdt, I have my x. I know my system is governed by some equation function, which can be very complex, nonlinear, multidimensional. But I assume there must be their function there, and I then there are some noise terms always, right? So if I can measure the deactivity of X, can we use as inverse problem? Can I learn what F is? Well, by the way, I want to mention this is again similar in physics, right? You do scattering. When you think about in the atomic physics, you do scattering. You measure all the things, then you try to reconstruct the potentials between the particles, right? Here you try to reconstruct the vector field. Yeah. By the way, this was mainly done by two young researchers, Xiaojie, the postdoc in Jonathan Weissman lab at MIT, and my student Yan Zhang. So what we do is, first of all, we thought, okay, we need, that's R velocity give you quite a reasonable estimation of the R velocity. I'm sorry, I have this allergy and couldn't find my drug. But we want to do better. So we developed a procedure as a machine learning procedure. Mainly, okay, we think about the gene expression of the gene transcription translation process, write down mathematical model. Then there's some parameters we want to utilize the single cell data to again, this is a machine learning problem, right? To learn those variable to parameters. So we developed those procedure and develop a package. We can do it. With that, then we want to determine the function. How do you do that? Well, since we do not know what the function form is, so we have the functional thing, right? We can determine what the best function. So we talk about a function space for the best one, called Hilbert space. So if you have taken quantum mechanics, you may have touched on this Hilbert space. If not, just let's use simple explanation. Let's say I have a set of data, X and Y. I want to fit. What I can do? I can do linear fit. I do linear fit. So I have constant term, linear term. Then I try to get what's the best fit. Give me so I can determine those parameters A0, A1. Okay. I can do more one. So it's maybe not a linear. I have this nonlinear terms. I can use one constant x squared, even higher. So it's kind of a polynomial. So I can still do the fit. Try to get what's best. So you see, this is the right thing give essentially form a basis set, and this basis not necessarily to be polynomial, right? You can go be essentially any other function. You have the basis, so you can write down basis function. Then any function can be expressed as a linear function combination of those basis function. So now the question is, okay, you try to determine those coefficients. So that's the basic idea. How the strategy will take. So the procedure is we have the data, very sparse, noisy data, have this X and have its deactivity. Then we want to learn a function that's a linear combination, have some basis function. So we have this procedure to do it. Then become technical. You do not worry about the details, but we got right. And the end, we have an analytical form of the vector field in high-dimensional space. Usually we do is it's a PC space. The reason is we know for a dynamic system, because a lot of constraints. Usually a dynamic system may be a high dimension, but only move along a low-dimensional manifold. That's allow us. We can only use a relatively smaller degree of freedom to describe dynamics. So we notice about 20, 30 PC modes already good enough. But those are really combination of many genes. Okay, then we can do a lot of analysis. Like here, we have as this toy model, we have the vector field. We can, there's some special regions called attractors. You see, you start from anywhere, then the system will go, right? Well, we'll try to one of the attractors. This is stable attractors, or this unstable. This is saddle points. right? So to say, so we can identify those things. So that's a toy model, but this is real data. So this is zebrafish post-embryonic development. So zebrafish development to different cell types. We reconstruct the vector field. You see, got those attractors corresponding to stable phenotypes. So we have vector field. What's a vector field? Vector field shows you if a system starting at a certain point and where to go next. So if we integrate them together, then we got the streamlines, right? The trajectories. If you start from another point, we got another trajectory. Then we can run simulations. I show here the zebrafish one. We're starting from this progenitor, we run the simulation, we showed how those individual trajectory move in this high dimensional space. So we can also do some further analysis because I said the vector field tells us a lot of things about the regulation, the interactions, like the Jacobian. Where the Jacobian is essentially define this. So this toy model, the 2-gene toy model here. So what's this tell us? Tell us if I change gene 1, how it will affect the velocity of the velocity along this gene 2 direction, right? So then you see, okay, if you look at the toy model, say along this line, let's just especially look at, we can examine this Jacobian at any point. Since we can get analytical expression, we can do it easily. But let's read this along this line. It shows us, okay, if I have I fix my gene 2 expression level, but I move the gene 1 and see how that affects the velocity, right? So it shows this, first of all, for the gene 1 on the component on 1, and then see this is increase, then it's increase. Well, it reflects biology, that's X1 self-activation. Well, on the other hand, this Jacobian shows gene 1, increase gene 1 will decrease the velocity along gene 2. That reflects that the biology that X1 inhibit X2. So you see from here, we even got some biology. And we can even do further, called this acceleration and the curvature. So this is differential geometry widely used in soft matter physics. Try to have to understand things. I myself learned some basics from this very nice review. If you can find out in context of soft matter physics. But here we essentially borrow it to study this vector field. So we see the acceleration, just as you know it, right? You understand it. So just how velocity change over time, and again you can calculate all those analytically. The curvature give you the trajectory, see the curvature, right? Band. So you can intuitively, what you can think about is you drive a car, then at a certain point you want to manipulate, you want to change the car state. You can either press the gas, press the brake, or turn your steering wheel. So that one you will change either both the speed or both the speed and also the direction the car will move. So so this can capture the time, the instance when you try to control the cell, control the car, while here control the cell, the genes can. So it's this happens early stage, even before the speed change significantly. So show the example, the cell. So here go you the toy model. I show you this is the acceleration field, and now you see there's a region has a very high acceleration magnitude, and you see what happens is you realize this is the region where you see the component shows you first of all this component, right? You see the acceleration vector along this direction here, and this component reflect X2, activate X2, the self-activation, and this one, right? You see X1 direction decreasing. So it reflects the fact that X2 inhibits X1. So biologically, what happens is at this region, because initially the cell have very low concentration expression of X1, both X1 and X2. Now they start to accumulate. When they start accumulate, when reach this region, so X2 reach see the threshold. So it's the threshold that can then self-activation and is inhibit X1 start to show become significant. So that's where the point is, and you can see show here is if you look at the trajectory over time. So you see initially just accumulate, then here is a dramatic you see acceleration. But the actual expression level change happens much later. So you see, if you only look at expression, you can only detect it much later time. But acceleration can show you what happens at early time. And here show you example. This again, the human A549 cells treated with a drug, then reduce the gene expression. So we can show, this is the change in reduced dimension. We show how it changes. Show essentially you have cell cycle, you have circle, then you also have other change to move forward. And show you this again. You see the Jacobian. This is Jacobian. Show you these two genes. You see the positive means okay. This gene ZhongB will activate SERPIN1. That's another gene. And here we plot the same data. We plotted at the gene space. So what I show here is see when ZhongB exceeds certain value, then its activation on SERPIN1 start to show up. Well, here why it's go down is because it becomes saturated, right? Then you increase further ZhongB, then the effect got saturated, won't increase again more further. So this is clearly show you we can from the data we can both get the biology and also quantitative information how the gene regulated another gene. Then we got this gene interact each other. We got the form kind of a network to interact. But here show you in that example that it's pancreatic endocrinogenesis. Again, it's a developmental process how the pancreas develop into different cell types. And there we also checked there are two pair of genes. We noticed the kind of the Jacobian kind of negative. And we know the two genes, indeed, the two genes kind of mutually inhibit each other. And in this progenitor region, that's where the two genes will determine which lineage it goes. So again, you see, by looking at the data, but this is from this dynamical perspective, you got a lot of information about the mechanism, right, quantitative information. So here we show, this again, the pancreatic data. We see, okay, from the Jacobian, we see what are the genes that have top Jacobian being top regulated. Indeed, those are the genes that determine which phenotype it goes. So here I want to show you another thing about acceleration. So here's that zebrafish. I show you this is the gene expression have different phenotype, and we look at the acceleration. You see this is acceleration, and here's a region you have a high acceleration magnitude, and we know, for example, right? So here the cells will differentiate into this cell type eventually, and you see this acceleration happens before it's fully commit to the cell type, as I just mentioned, right? This is acceleration. Reviews the early event, and that's exactly what we found out. Although the genes that contribute acceleration, indeed, those genes that they direct those cells go to specific cell type. Interesting. Let's look at this one. So there's a phenotype in the original study. The researchers couldn't identify what are those cells. So we look at the acceleration. And because we say, okay, it might be those genes that try to direct cells go to certain phenotype, and we look at their genes, see what are the functions of the genes. Now, okay, they call it cartilage chondrocyte. So that's appear frequently. So that suggests those unknown cell types will be related to chondrocyte. You see, we have the analysis have predict power. So this is the FF49 cell undergo EMT, as I mentioned. We also have the data. We do analysis. Now we both have the snapshot. We also have this this velocity. We've got this kind of a vector field, so we can run simulations to trajectory. Interestingly, also found two types of trajectories. Well, remember in lifestyle imaging, we got two types of trajectory. So are they the same, corresponding to each other? So this is a very open question where my postdoc, Doughty and Ke, and they try to work on it. So the idea is, okay, we have single cell data. We predict there are certain two trajectories. Then experimentally, lifestyle imaging, we measure two trajectories. Their correspondence. So that's we need to do this lifestyle imaging plus study its gene expression, and so this is still ongoing research. But what I hope I can show you, I know it's kind of a rich lot of biology. Some topic may not be familiar to you, but I hope I have described all those things in the language of physics, right? So try to use a language you're familiar with, and we see we can extract lots of information from this single cell data, and that's really parallel to what happened in physics, right? When we study both the planet motion and the atomic level, the particle interactions. So that's where I suggest. I said this is a cell phenotype transition dynamic studies are really exciting frontiers. Both for cell biology, right? So because this is very fundamental to understand the life, but also they are, I think, the prototype for non-equilibrium physics, and they're very exciting physics will come out. They're very exciting phenomena. Then it's also open, more open for experimental lifestyle quantitative study. Well, can feed back to theoretical studies. So with this, I want to also mention people who do their work. I already mentioned some of them during my talk, and also this is all collaborative. We're collaborating with many colleagues like Jonathan Weissman Lab, Yvette Bahar, my colleague, and also my colleague Simon. Many other people, also funding agencies. Yeah, before I close, I also want to do this kind of advertisement because Pretam mentioned, we discussed on education. I want to mention, in our University of Pittsburgh, we have many interdisciplinary programs, and also traditional thing for graduate studies. So this is one program is called Joint CMU Pitt Computational Biology Program. We recruit people from different backgrounds. Some people say a computer science background, but also here we strongly encourage people with a physics background to go. This year we do have students from India, and her background is just physics. She has a double major in physics and biology. And also, I'm also affiliated in this physics astronomy program. There, lots of people can look at. I think with this, I'll stop here and take questions.
Yeah, thank you, sir, for your wonderful presentation. So we have got a few questions in inbox. If you allow, we can start our discussion session, sir.
Yes.
Okay, so first question about the epithelial. So what are the effect of quasi-potential on epithelial?
What
are the effects of quasi-potential on epithelial?
Okay. Yeah, that's a good question. So here we make the attempt to understand this process from physical perspective, and there the quasi-potential I think you can still see my slide, right? So there we want to, because we know the track from dynamic system theory, we view as the cell phenotype just a tractor, right? If it's a conservative system, then you can write down potential. That's the potential well. So we essentially try to utilize those kind of picture to quantify this process. So what happens is, without TGI beta, we know the cells mainly stay in this region that correspond to epithelial cells. There are always fluctuation. It's not thermal fluctuation, but there's all this gene expression, all things kind of stochastic. The environment have some random noise, so there's a fluctuation, but mainly located in this region. Then TGI beta does is so you see this is make this when you add TGI beta, because TGI beta will interact with all the genes or things that generates a new potential. So then the system you can view as a kind of evolve along this potential. Then from our physics language, it was like a relaxation to a new attractor. So I try to use the physics language to explain this why TGI beta treatment induce this spontaneous EMT. So I hope that can answer the question. But I always say, I want to with this one, but always keep in mind, we are talking about non-equilibrium system. So I try to use this potential as kind of help understand, but there are limitations, right? Yeah. Right. There's sense because we know the system dynamic cannot be fully described by a scalar potential. So that's why in the second part we talk about the vector field, which is a more complete description of system dynamics.
Yeah, thank you, sir. We have another question. So, what is the post-embryonic development? I think you have already explained about, but
the post-embryonic development.
Yeah.
Okay. Yeah. That's so this kind of kind of biology thing. First you have the fertilized egg, then develop into embryo. Then we so for the deeper fish, right? Then develop into this like more like a fish, right? Thing. So it's really biology. They want to decide what's the try to divide to different stages. I couldn't remember exactly how they. It's really when the embryo has reached certain size, then they no longer call it embryo. They call it post-embryo development. There's another stage which I couldn't remember. I forgot what they call it.
Yes, sir. So this may be the last question. So how can cell fate regulate or guide phenotype control?
How cell phenotype?
Yeah, cell fate regulate phenotype control.
Oh, so cell fate. We know cells can assume different phenotypes. I mentioned, right? There's skin cells, neurons, all things. That's one thing. And those even can go go death or thing, right? So all those are tightly regulated by that of genes, the programs. This is very fundamental questions. How we want to understand how those genes interact with each other, regulate each other. So this is like computer program, right? If you know computer program, we have all those instructions tell what computer do. So our cell have built in those program. We really want to understand what is this program. So this is kind of reverse engineering, right? So this we know how they work together. Then we first of all we understand how the cell control their cell fate to determine if the cell receive a signal. It always need to make a decision. Does I need to stay here, or I want to perform apoptosis, suicide, or I need to go some other things. So this this is very important for developmental process that then we can develop into the right organism. But then for those drug for those disease like cancer. The reason for the cancer development is that for those cells, they something goes wrong. Then the normal cell, when things goes wrong, so and go apoptosis, kill yourself. So then there the healthy cell can replenish. But those cancer cells, they somehow they have the mechanism to avoid this built-in program to instruct it or to undergo apoptosis, but rather they can survive, right? So then from us also, how can we correct this program? Really, okay, still tell the cancer cells you need to die, right? So this is the important problem in biomedical research. But you can see this is from theoretical perspective also very interesting.
Okay, sir. So thank you for your wonderful presentation and discussion session, sir. So I think our students have learned a lot of things about the chemical physics, and as you have already explained that the physics student can also join in this field. So could you say something for our student if anybody want to do their higher study in chemical physics? So what he or she need to do? Could you say something?
Yeah, thank you. For sure, I think I talked to Pretam. I think he made a lot of efforts try to broaden you guys, the scope. I think he made very important, efforts. You guys, you are lucky to have him as your instructor. I think interdisciplinary research now becomes a really trend. Right, but my suggestion is make your grounds as solid as possible, because I do know that some people we have even for undergraduate research or undergraduate program we have an interdisciplinary program versus a more traditional physics chemistry thing. My feeling is I always probably the best is that you guys have a solid training, in the graduate level. You can expose then to make transition, which relative because you have solid ground, right? In terms of chemical physics thing, I think so. I'm not sure. Chemical physics you both been touched in physics department and chemistry. And I have, I said I have a postdoc now in India, IT, and he also the background of, this chemical physics. So so I'm not sure it's in your place how in chemistry department, in physics department there's something related, but that's the underlying a lot of methodology all kind of common, right? You can see chemical physics somewhat also closely related to condensed matter physics or statistical physics, right? Yeah, but just if you guys have a student question or thing, feel free. I'm very happy to answer. If you send me email things, anything I can help. Embassy. We in our program we have a lot of international students. Each year we have many from India as well. So I think in the fall I'll most likely host 2 students to do rotation project in my lab from India. So really, it's Indian students very work very hard, have very solid background. Really, there are a lot of opportunities. I would like you guys to explore.
Thank you, sir. So as the video will remain in YouTube and Facebook, so anybody, if they want to ask question, they may write there, and also they may send you the email. So to you.
Yes.
Thanks for accepting our invitation and for giving me opportunity to arrange such an important webinar with you. So it's my pleasure and honor to host you in my physics webinar, sir. So bye for today, and hopefully in near future we will arrange another webinar with you if you have time.
Yeah,
so bye
that would be happy.
Yeah, have a nice day.